our next speaker is a person that I've never personally met, but who I am in awe of because of what she has done in being able to miniaturize things and have an effect of using various chemical sciences techniques to really affect human health, from the use of gold nanorods to doing such things as creating micro livers. And I'm very pleased that we have a chance to hear from Sangeeta Bhatia, who's at MIT.
Hi, thank you. Try and share my screen here. Let's see.
You got it.
Yeah, great. Well, I wanted to thank the foundation for inviting me today and give my huge congratulations to Bob for this wonderful prize. I'm going to focus my remarks today on the intersection of miniaturization technologies and medicine in cancer by focusing on cancer nanotechnology. And that's in part to honor the 50th anniversary of the National Cancer Act, which was passed in 1971, you may know that, which led to a coordinated effort to reduce cancer death rates. And in 1992, that actually happened. Cancer death rates started going down. And that is the year I met Bob Langer. And I was entering graduate school. He was a visionary and a pioneer even then, but I'm lucky enough to now call him a colleague and a friend and a mentor. So congratulations, Bob. And as I said, I'm going to tell you about our work in cancer nanotechnology, much of which he's contributed to. These are my required disclosures. So just to sort of set the stage, I'm an engineer and a physician by training. I use chemical tools in our work, as you'll see today. And one thing that really inspires me as an engineer is the power of miniaturization. So this image is to show you that one transistor, where one transistor used to fit, we can now fit 1 billion. So this has really revolutionized computation and made it portable and had a transformative effect on many of our lives. If you sort of think about that in the arc of history, what is it that brought us to that point? How did we get so small? You know, it starts in the 60s with the mainframe computer and then IBM. And then finally we break the sort of nanotechnology length scale 100 nanometer limit in the year 2000, which is when I started my lab. And now we're really well under 5 nanometers in recent times. So it's been an incredibly powerful push to miniaturize technology. And what's really fascinating about this length scale is, of course, that at this tiny length scale, at this sort of single nanometer length scale, that the physics of materials actually changes. And so here on the right are a whole host of materials that many of you are familiar with, which exhibit these nanoscale physical properties. So this has been very exciting for someone who's an engineer. And what's even more exciting is that the biological properties of these materials also changes at this length scale. So for example, where they will traffic systemically is size-dependent phenomenon. Whether something goes into the liver or out of the kidney is in part a size-dependent phenomenon. And so what we've been really interested in is how one can combine nanoscale physics and nanoscale biology for applications in medicine. So the stories I'm going to tell you are two. I'm going to tell you a diagnostic story and a therapeutic story. And as I said, in the context of cancer. And just to sort of set the stage, this is the landscape of sort of a typical patient journey in cancer. And so you'd like to be able to assess risk early, detect disease as early as possible, diagnose that disease, make an intervention, monitor the treatment, and then monitor for recurrence. So this is sort of the whole landscape. And we know that if we can find cancer earlier, that we will have better outcomes. This is just a recent plot showing you for a number of solid organs that are found when they're localized, the 5-year survival is much increased. And today we actually looked for most of these cancers largely by imaging techniques. So you hear CT scans and pap smears that are seen on a microscope. And more, there's a new field emerging of so-called liquid biopsy. So now we are looking in the bloodstream for shed analytes that come directly from tumors. And these can be metabolites or proteins or exosomes, or most excitingly recently, cell-free nucleic acids themselves. So this is an emerging field that's very exciting, and really the challenge for physicians is finding tools that are both sensitive to that early cancer and specific. And you know, the challenge with the liquid biopsy field, however, is that you can only measure in the blood what the cancer sheds. That's the fundamental limit. And so what we've been thinking about is whether there are nanomaterial designs that take advantage of those size-dependent switches I alluded to earlier that could add molecular analytes into the system, create molecular analytes that never existed in the body. And we call these synthetic biomarkers. So I'm going to show you a movie. This is a cartoon, and I'll show you chemistry in a second, where we've made a nanomaterial that's 100 nanometers in size. We inject it in the bloodstream. And here you can see it's extravasating from that blood vessel into a surrounding solid tumor through permeable vasculature. And those holes are about 100 to 200 nanometers in size. So these materials can now access the tumor interstitium. And there we design them to be vulnerable to cleavage by tumor-associated enzymes, for example, the matrix metalloproteinases. And of course, one copy of an enzyme can cut many, many bonds in an hour, and so we get an amplification effect. So when these materials are acted on by these proteases, they liberate a reporter. And that reporter is actually made chemically so that it won't degrade in the circulation. So that's made of D-amino acids in this case. It's lovely to talk after Peter Schultz about this. They re-enter the circulation, and then the kidney is a 5 nanometer filter. So if these fragments are smaller than that filtration limit, they will be concentrated into the urine. So the way this whole system plays out is you can inject this 100 nanometer size particle systemically. It will interrogate the tumor and be activated, catalytically amplify these reporters, and an hour later, they accumulate in the urine. So it's like a non-invasive way to generate molecular analytes by interrogating a tumor deep inside the body. That's the way the system works, and we have been playing with the chemistry of this system to enable different clinical use cases. So I'll walk you through some of those. I already alluded to the class of enzymes that we've been interested in studying, and that's the proteases, which is a lovely large family of proteases, and they have been long reported to be involved with cancer pathogenesis. So virtually at every stage of cancer development, growth, survival, the so-called angiogenic switch when blood vessels are recruited into cancer, and invasion, metastasis, and importantly, interaction with the immune system, which is more and more important. So all of these things are associated proteases that one could make sensors against. Okay, so what were the cartoon character sensors that I alluded to earlier? This was our first generation material that we published actually back in 2008. And at the time, these were iron oxide materials that were dextran coated, so they're super paramagnetic materials. And covalently coupled onto that material was a tandem peptide. So a cleavable amino acid sequence and then a reporter sequence, in this case carrying a fluorophore. And so you can see this material, when interacting with thrombin, is thrombin sensitive. And this quenched fluorescence is dequenched upon cleavage. So this is a fluorescence on sensor, and you can see that it activates very rapidly in the presence of thrombin and not other proteases. If you make the same material out of a D-amino acid version, it will not activate in the presence of thrombin. And if you make the L-amino acid version and you add a thrombin inhibitor, it also doesn't activate. So you can make these sensors activate in the presence of different proteases. And essentially what we did in our laboratory was make libraries of these materials. We've now made a few hundred versions of these and couple on their surface different amino acid sequences and characterize them here for catalytic activity against recombinant proteases. And we can make these heat maps. So here's, for example, thrombin, which I showed you in the last slide. So this is the amplification sensor piece of the system. And so then the next piece of the system is the size-dependent trafficking that I told you about. So here's that experiment. So here now we've made the same material susceptible to MMP2, so a type 4 collagenase. And what we've done is administer it to a mouse that has liver fibrosis, where it's making this type 4 collagenase, versus a control. And you can see that the materials accumulate in both cases in the liver. However, when the mouse has MMP2 in the liver, it acts on that material, it liberates, in this case, a fluorescent reporter. And after an hour, what we see is this second organ lighting up, and that's actually the bladder. So that's the urine in the bladder that's now fluorescent. So this is how we actually first discovered this system. And, I started my talk by saying that sensitivity and specificity are both important. And so if we really just have one analyte, it's hard to make something both sensitive and specific. You're always trading off. And the solution to that, as you all know, is multiplexing. So if you make many measurements, then one can build a classifier. And so the way we multiplex this system was by using mass bar-coded reporters. And so now this tandem peptide that I showed you before has an L-amino acid domain that can be cleaved by a protease of interest, and then a D-amino acid domain so that it won't be degraded in the circulation, and it can carry a unique mass. We can confer a unique mass by introducing heavy isotopes into this. So you essentially have a barcode library. So here's a 10-plex. When it fragments on mass spec, you can see the barcode emerge. So now what we can do is administer a cocktail of these materials. And so here, for example, you see a cocktail of materials that are entering into the liver, all interrogating the microenvironment. Those reporters are re-entering the circulation and coming out in the urine in an hour. And you would mass spec the urine to read out the signature. So this is an idea that we first published in 2013, and we have been advancing. It's now in the clinic through a startup company, and it's been through many, many different formulations. And so the current formulation has now at its core an inert polymer, polyethylene glycol, and it has the chemistry that I see here on the left. And we've characterized it extensively in a number of diseases. So we've shown for cancer detection that it's a highly sensitive system. We have that amplification process of the cleavage event. We also have the concentration of the kidney. It's about a 20-fold concentration from the blood to the urine. The limit of detection in mice is a 3 cubic millimeter tumor, so it's pretty tiny. The specificity, as I said, we achieved through multiplexing, and so far we've done a 19-plex version of this tool. And in mice, which are all isogenic, so all identical genetically, you can classify mice with a 3 to 5 probes, so perfectly. And all the extra plexes are to go into humans that are more noisy. But just to show you an example of the kind of data that we get, we mass spec the urine, we put it through a machine learning algorithm, and in this lung cancer example for early lung cancer detection in mice, two mouse models of lung cancer, you can see almost perfect classification using this non-invasive tool by collecting the urine at an hour. The fun part about this as engineers has been actually to think about these different clinical use cases then and build off of this system. It's a completely synthetic system, and you are creating the analytes that will be useful to the physician. So we've been thinking about diseases like cancer, as I said, but also diseases like fibrosis, where proteases are involved, thrombosis, so blood clotting, and then infectious disease, which also involves lots of immune cell associated proteases. I mentioned to you that this is now in the clinic, so it was really interesting to get this actually through the FDA because it's an injectable product with a diagnostic readout. So for those of you who are interested in that, it would be fun to talk about it during the question section. So just to sort of give you an example of the type of clinical use cases that one can then use the chemistry to drive, one could be, what about a point of care version of this? So it's all well and good that we have a mass spec urinary readout that's highly sensitive and specific, but what about something at the point of care? And so here you can now change the reporters, so the D-amino acid domain, so that they have a biotin capture tag and a small molecule ligand, and design them so that they could be captured on paper. So this is now a lateral flow assay, like a pregnancy test, where you would essentially do a urine dipstick for a readout. So this is one use case that we've done using it this way. We also have a CRISPR version of this lateral flow assay readout. Another use case might be that you want something much more rapid than the one hour urine test. And here we were actually challenged by the Gates Foundation to think about TB detection in the lung in patients that might be dehydrated. And so here what we did was actually re-engineer this particle again so that you have that same PEG core, the same cleavable peptide linker, and now the reporter is a volatile organic. So here, this is a hydrofluoramine, and we have a breathalyzer. And this breathalyzer peaks at about 10 minutes. So you breathe it in and you breathe it out, and you're detecting that cleavage event in the breath. You're creating a volatile for which there is no breath background. Finally, then, one might imagine that once you've made a cancer diagnosis, the physician would like to localize the cancer for a follow-on therapy. And so here what we've done, this paper is about to come out in Nature Materials, is make a version of the same probe that can be loaded with copper-64. And so you can image this using PET imaging. And so here you can see in this mouse model of lung cancer that we could actually image the lung cancer using the same version of this that gave the cleavage reporter in the urine. You load it with copper-64, and then you can visualize the tumors for intervention. So this just gives you an idea of what kinds of flexibility this sort of synthetic biomarker concept can afford you. And I'd like to now turn my comments to the ideas of cancer nanotechnology in the therapeutic domain. So once we've made a diagnosis for which we'd like to make an intervention, what can one do? And of course, on the bottom here are really the mainstays today of cancer therapy: chemotherapy, radiation, targeted small molecules, excitingly immunotherapy, and even cells that can be weaponized, so-called CAR T cells. This has been a very active area of research. And if one thinks about what else one might be able to add to this picture, in this moment, nucleic acid therapeutics offer a lot of promise. So if I give you that sort of same time scale that I showed you with the miniaturization technology earlier, look all the way back to the 1960s when we found mRNA, and RNA therapies have actually been steadily coming onto the radar. The first FDA-approved RNA therapy was actually in 1998. The first RNAi trial for cancer was in 2010. Alnylam's 2 approved products in 2018. And then of course, 2020, 2021 are the year of RNA in COVID-19 vaccines. And actually Bob had a great deal to do with that. So it's been a really exciting time for RNA, but nevertheless, there is still no RNA drug approved for cancer therapy. So in spite of all the promise of nucleic acid therapeutics, what about cancer as an indication for RNA? So what is the problem? One of the problems is, of course, that RNA doesn't penetrate into tumors. So here's just a picture with a small molecule where you can see in this mouse model of pancreatic cancer that it has been infused with FITC, that this molecule is not penetrating into tumors. This is the so-called interstitial problem of tumor penetration. And that's not just true of the primary, but even the metastatic tumors. It physically keeps some small molecules out. And of course, for RNA, given its charge and physicochemical properties and susceptibility to degradation in the circulation, RNA is even worse. So that's at least one of the problems, is tumor transport. So what we've done in collaboration with Erkki Vuoslahti over the years is come up with a nanocarrier system that's made of a different tandem peptide that can complex with siRNA and form a particle that can actively penetrate tumors. So I'll show you a cartoon first again, and then the chemistry later. So the carrier here has cyclic peptides that target the tumor and activate a nutrient transport pathway. It has another peptide that's called transportan, which is an amphipathic endosomal escape peptide that helps get the RNA out of the endosome once it's in the cell of interest. It's also cationic and can interact with that RNA cargo and form a particle. So we have this carrier, it's about 60 nanometers in size. And the way it works, based on many, many years of work in Erke's lab, is shown here. So here you see this carrier traversing the vessel around which there's a tumor. And essentially what happens when it interacts with the cell membrane is there's a cleavage event that it gets activated by a cell-associated protease, UPA, and that cyclic peptide linearizes and exposes a C-terminal arginine. So here you can see it binding the integrin, linearizing, and that C-terminal arginine binds a receptor known as neuropilin-1, and neuropilin-1 activates an mTOR-dependent transport pathway. So that's a lot of biology, but basically what happens is this gets activated on the endothelium of tumor cells and activates a transport pathway, can get inside the tumor and then out of the endosome. It's important, obviously, to be in the cytoplasm where you can deliver, in this case, siRNA and silence a gene of interest. So how does this work from a chemistry perspective? So we make these materials out of tandem peptides I described. So this is an 8-member amino acid ring cyclic peptide called IRGD from Erke's papers. This is the endosomal escape peptide that I described, transportan, which is an amphipathic, pH-dependent endosomal escape peptide. And then this peptide is actually myristylated. It's inspired by envelope viruses that have this membrane interaction group. We mix this tandem peptide together with another tandem peptide that has polyethylene glycol in line in the chain to increase stability of the particles. And so using these two components, you can mix it with a nucleic acid, form a particle, and administer this systemically. And so here what you're looking at now is a tumor where the siRNA cargo is fluorescent, and you can see over time that it very robustly accumulates, extravasates from the vessels and accumulates in the tumor interstitium. And if you make the exact same material out of a version of that cyclic peptide that does not cleave and does not have that C-terminal arginine, you can see that it'll bind the endothelium, but it won't traverse, it won't penetrate into the tumor. So when we use these tumor-penetrating RNA delivery vehicles in mouse models of ovarian cancer, silenced a gene that was involved in ovarian cancer called ID4 that we discovered with colleagues at the Broad Institute, you can see a robust survival effect in these animals, and it's dependent both on the sequence of the SI, but also on this penetrating aspect. So finally, my last couple of slides I see Matt popping on here are what else could one potentially deliver with this nanomaterial? So now this is just a charge interaction. You can think about any nucleic acid essentially. And so we've done a couple of other examples of using this material set. So the first is in immunotherapy. So you can encapsulate immunostimulatory oligonucleotides. And we did that in this example. And then we're able to show in combination with a checkpoint inhibitor antibody that we could robustly activate the immune system in the presence of these tumor penetrating nanocarriers and treat cancer in mice. The second thing that we're doing is looking in pancreatic cancer. So here you're looking at a pancreatic tumor, again with fluorescently labeled siRNA against an oncogene called KRAS and delivering that deep into the tumor in pancreatic cancer. And finally, in an infectious disease moment, it's been really exciting to see coming full circle back to silicon, which I started with, that you can actually use porous silicon, load it with siRNA, and use the same kind of delivery strategy to silence the inflammatory processes associated with infectious pneumonia. And this was work done in collaboration with Michael Saylor. And so this looks a lot like the cytokine storm, for example, in COVID. So these materials, I believe, can really help us carry RNA into hard to reach disease tissues to manipulate the biology at the cell of interest. So if I just sort of reflect on what's brought us here, there's these two time scales that I showed you, miniaturization and nucleic acid therapeutics coming together over these decades. And of course, they've been supplemented by many, many other fields that are now all converging on what I think is a really bright future for cancer research, where we can detect cancer earlier, we can treat hard to treat things, we can hopefully address metastatic disease. And I'm personally really interested in cancer care kind of around the world. The last thing I'll say is that you should come visit us post-pandemic at MIT. Bob and I are in the Koch Institute, where I run this center, and the center is actually founded on the NCI-funded Cancer Nanotechnology Center that Bob started, and we welcome chemists in the conversation on the role of nanotechnology in cancer. And we are hard at work trying to train the next generation of interdisciplinary thinkers to reimagine what the future of cancer care will look like. So with that, I'll thank you for your attention, and most of all, Bob, for his mentorship and friendship, and congratulations again.
Thank you, Sangeeta. I was myself a little bit uncertain about how you actually penetrate the cell, and it seems so important. Is it one arginine or a string of arginines that actually help you get through the cell? What is going on in terms of the penetration into the cell of your nanoparticle?
Yeah, it's a great question. So the motif is RXXR with the R on the end.
I see. Okay. Now there's another question in chat. Let's see.
Oh, I can't see the
No,
chat. Let me see.
no, I've looked. No. Well, thank you again. And it's actually very hopeful what you're saying about really seeing how medicine is being, I think, revolutionized by the application of these types of concepts coming together, highly interdisciplinary, but certainly chemical sciences playing such an important role too.